So in this talk, we're going to be working on the cell membrane, and this is chapter 7.3, but also 7.4 in your Miller and Levine biology textbook. So please make sure that you're reading those sections there. Make sure that you're reading those chapters. Anything to do with the membrane is fair game. So what is the function of the cell membrane? Well, first and foremost, its job is to separate the cytoplasm from the extracellular environment. So the cytoplasm is the inside of the cell, and the extracellular environment is the outside of the cell. The second thing that it does is it controls what goes in and out of those, what goes in and out of the cell. And it does that by these things called transport proteins. The cell membrane also has a few other jobs here. They're more minor jobs, but they are still jobs. So one thing it can do is that it can carry out chemical reactions using enzymes. It can identify what kind of cell it is using different types of antigens, which are kind of like the fingerprint of the cell. So we write that in there. Antigens are kind of the fingerprint of the cell. And so that will help identify what kind of cell it is. And it can receive signals from other chemical messengers, and that's done by receptor proteins. Now, just as a little reminder, the cell membrane can also be called the phospholipid bilayer or the plasma membrane. So the cell membrane can also be called the plasma membrane. And it's made of a phospholipid bilayer. Remember that a bilayer is a double layer. And so, yeah, we can see here that the cell membrane is separating the inside from the outside. It's made of a phospholipid bilayer, and it's going to control what goes in and out of the what's going in and out of the cell. So we've talked about this already, but we'll just do a little review here. The cell membrane is arranged in a continuous double layer, aka the phospholipid bilayer, of phospholipids, right? So it's got these phospholipids. Over here is the diagram of a phospholipid. Remember, we've got a few parts. We've got the phosphate, We've got the glycerol backbone here. So here's the glycerol backbone. Here's the phosphate head. And you can see just look how many oxygens there are. Oxygen, 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 oxygen. So there's all these oxygens, and what that does is that it pulls the electrons. The electrons get pulled up towards this side here. So that makes this side up here, this part of the phospholipid is hydrophilic, or in other words, it's polar up here. And then down here, this part down here is all non-polar. This is the non-polar part of the phospholipid. So because of that, it makes a phospholipid bilayer. And so remember that the hydrophilic or the polar phosphate heads and the glycerol backbones, they face the watery environments. So that's going to be the extracellular environment and the cytoplasm. And then the hydrophobic fatty acid tails, right? These parts right here. Let's do it in orange. These parts right here, these fatty acid tails, they are non-polar. They are going to face the interior of the phospholipid bilayer, and it's going to create a waterless middle, or a waterless middle, a waterless interior. Maybe a better word would be interior. right? So the middle portion of the phospholipid bilayer does not contain water. It's afraid of water. It's hydrophobic. Now, there's other molecules that are part of the cell membrane, and we'll talk more specifically about these, but proteins, glycoproteins, glycolipids, and cholesterol. Now, glycoprotein is a protein and a carbohydrate together, whereas a lipid is a lipid and a carbohydrate together. And then cholesterol, remember, that's one of those lipid, that comes from one of those lipids that that is the precursor to a lot of hormones. And cholesterol is often thought of as being bad, like a bad fat, a bad type of lipid. But that's not actually true, right? It's not actually true, because it's actually important for our cell membranes. Too much, yes, but we definitely need some. It's not fair to say it's bad 100% of the time. And then we've already talked about how the phospholipid bilayer is semi-permeable, or we talked about how it's selectively permeable. It only lets some substances in and out. It controls what goes in and out of the cell. So the phospholipid bilayer is not just made of phospholipids. It's actually made of lots of different things. And it's not a fixed, it's not a solid structure. It's like a semi-liquid. And so it is very movable, it's very flexible, and there's lots of different stuff that's stuck into it. So the word fluid means that it's moving. It's always moving. And the word mosaic means that it's a mixture of things. So you can see here, we've said that already, right? It's a mixture of things. And so it's made of many different types of macromolecules. And some of those macromolecules include phospholipids, obviously. There's proteins that are stuck into the cell membrane, or that are on just the surface of the cell membrane. Glycoproteins and glycolipids. So here we have, this right here is a glycoprotein. This right here is a glycolipid. That says GL. This right here is a glycolipid. And then cholesterol. And cholesterol is this stuff right here. You can see cholesterol is labeled right there. And so this molecule here, these molecules here are cholesterol. And you can see there's more cholesterol right here, right here, right here. So these are all cholesterol molecules. And we'll talk about the function of each of those. I would recommend that you go and check out the Amoeba Sisters video here. They do a really good job illustrating this and animating that. Definitely go check that out. Membrane fluidity. So like I said, the membrane is flexible and it's always moving. And there's two ways that we can control that. We can control that with phospholipid tails. So our cells can control their fluidity with the structure of their phospholipid tails, or they can control it with cholesterol. So let's talk a little bit about these two things. So the problem that cell membranes have is that as temperature increases, they become more liquid. They become more fluid. So what happens is that the contents of the cell can actually leak out of the cell. And so as the temperature of the cell membrane increases, it needs to find a way to maintain become a little bit more solid. Now, on the opposite side of that, as the temperature decreases, the membrane becomes too solid, right? And then it loses its flexibility, right? It's too rigid. It's too rigid. and it needs to be flexible. So we can't have it lose its flexibility. So the problem is increasing temperatures, it becomes too fluid. Decreasing temperatures, it becomes too solid and not flexible enough. So the cell has these enzymes in the membrane that actually are able to change the fatty acid tails. So if we look here, here's the fatty acid tails down here, right? And if we go back to this picture here, I think I've said that the fatty acid tails can be saturated or they can be unsaturated, and it's not every phospholipid always has one saturated and one unsaturated. They could both be saturated, they could both be unsaturated, depending on the condition of the cell. And so if the conditions are too warm, then that means that the membrane is starting to become too leaky, it's becoming too fluid. And so what's going to happen is that as the temperature increases, the enzymes in the cell are actually going to make more saturated fatty acids in the phospholipids. And what this is going to do is it's going to allow them to pack more tightly together. You can see that they're able to squeeze together tighter here, right? So they're essentially, they are more tightly packed. But if the temperature decreases and it gets too cold, the environment of the cell membrane is too cold, then what the cell can do is it can make more unsaturated fatty acids. And what this does in turn is that it increases the fluidity, right? Because now there's more space. So these phospholipids are able to spread out a little bit more. And so there's more space here. So that way it's able to maintain its fluidity, so it stays more flexible. The other way that the cell membrane does this is with cholesterol. And cholesterol is kind of like, you can think about it as like the buffer of flexibility or fluidity in the cell membrane. So in high temperatures, it helps the phospholipids stay together. It helps them not fall apart, essentially. So it keeps them together. And in low temperatures, it provides kind of a spacer between the phospholipids. So it's essentially just buffering the flexibility of the cell membrane. A video to go and check out is on Khan Academy. It definitely Mr. Khan and his team explain how this works in more detail there as well. Okay, now we've talked about how the cell membrane is a mosaic of different things. It's made of lots of pieces. And so we know that it's made of phospholipids, but it's also made of proteins. And the proteins in the membrane have all sorts of different jobs, but there's two general types. There's integral, or we can also call them transmembrane proteins. They go across the cell membrane. So here's an example of an integral or a transmembrane protein. It goes all the way across from one side of the cell membrane to the other side of the cell membrane. So part of it is touching the extracellular environment, part of it is touching the cytoplasm. And some of the things that these things do is that they can transport materials across the cell membrane, or they can help cells stick together to form tissues. Okay, let's just go like this. Sorry. The other type of protein is called a peripheral protein, and peripheral means it's to the side, so it's just on one side of the membrane. And so you can see here's a peripheral protein here, and then here's another peripheral protein here. Sometimes like this one here is kind of stuck into one side of the membrane. That happens. Sometimes it's just on the surface of the membrane like this one down here. But their jobs are to connect the cytoskeleton to the cell membrane as like one of the main jobs. They have other jobs, but that's kind of one of the main ones. So here's some other jobs of proteins. They can be transport proteins like we talked about. They can help with cytoskeleton attachment like we talked about. Here's that peripheral protein there. They can stick cells together. There's those two transmembrane proteins. Here's one cell and here's another cell, and they're sticking together. But also here's an antigen here. And so these glycoproteins, the carbohydrate part, this is called the antigen. And it's kind of like it identifies the cells, right? And it's made of carbohydrate. And so because we've got a carbohydrate and a protein, this is called a glycoprotein. Some proteins inside of the cell membrane are enzymes. Here's that enzyme's active site, and it's going to take this substrate and change it into a product. Some of them are receptors. So we talked about, like, this could be a neurotransmitter here sticking into a receptor protein, and then some sort of cell response will happen as a result of that signal. So this is kind of like the signal. This is the receptor. And then the signal will attach to the receptor, and the receptor will start a cellular response. Okay, so we did talk about this just a second ago. Carbohydrates can stick to proteins, or they can stick to lipids. So whenever you have a protein with a carbohydrate, we call that a glycoprotein. And when you have a lipid attached to a carbohydrate, we call that a glycolipid. So the word glyco equals sugar, or in other words, carbohydrate. Okay, so here you can see the carbohydrate chain attached to the protein there. So here we've got a glycoprotein, and then over here we've got a glycolipid. The carbohydrate, this is also a carbohydrate, is stuck to just a lipid.